Hello everyone, welcome back to Mahi's Life Science channel. In this video, we will discuss about the plastic biodegradation by microorganisms. Biodegradation of plastic occur in four important steps. First one is biodeterioration, second one is biofragmentation, and the third one is assimilation, and finally fourth one is mineralization. Biodeterioration of plastic, which involve both physical as well as that chemical biodeterioration. In this, superficial degradation that modify mechanical, physical as well as that chemical properties of the plastic. In most cases, abiotic parameters contribute to weaken these polymeric structure. Biodeterioration seems to be triggered by the formation of the microbial biofilm growing on the surface as well as the inside the plastic. Once microorganisms started to grow on the plastic, it start to secrete the extracellular enzyme. Also, it can able to adhere the enzyme to the plastic surface, which leads to the cleavage of the polymeric chain. Finally, erosion of plastic surface will occur that we called as a biodegradation. We can get the end product like carbon dioxide, water as well as the methane. Research studies showed that the biofilm of Rhodococcus ruber C208 formed on polyethylene showed high viability and even after 60 days of incubation adhered to the polyethylene without any supplementation of the external carbon. Physical deterioration of plastic. The formation of the microbial biofilm is associated to the secretion of extrapolymeric substances that we called as an EPS that reinforce the cohesion of the biofilm and the adhesion to the plastic surface. These EPS enter the pores. Also, microorganisms they can able to grow inside, thus increasing the pore size and provoking crack that weaken the physical properties of the plastic. Chemical deterioration of plastic. The microbial community that developed on plastic may have high diverse and the development of a biofilm may release acid compounds such as nitrous acid by Nitromonas species, nitric acid by Nitrobacter species or sulfuric acid by Thiobacillus species. Mainly, these chemical deterioration can take place by chemolithotrophic bacteria. After that, organic acids such as oxalic, citric, fumaric, gluconic, glutaric, glyoxylic, oxalic, and oxaloacetic acid may also be released by chemoorganotrophic communities. The pH inside the pore is then modified, resulting in a progressive degradation that changes the microstructure of the plastic matrix. Biofragmentation. The fragmentation of plastic polymer into oligo and monomers can be various origin. Example, mechanical, UV radiation, thermal, chemical or biological method. Plastic polymer or molecule with high molecular weight that cannot cross the cell wall. Microorganisms secrete extracellular enzyme that we call as an exoenzyme that can catalyze reaction principally at the boundaries of the plastic polymer. They can perform many chemical reactions, but they generally need imbalance of electric charge to perform lysis. The main limit of biofragmentation is the stability of the plastic polymer, which are constituted by a long chain of carbon and hydrogen that contain very balanced charges. To destabilize the local electric charge, bacteria can break down plastic usually contain enzyme called oxygenase, which can add oxygen to a long carbon chain. For instance, monooxygenase and dioxygenase incorporate respectively 1 and 2 oxygen atom forming alcohol or peroxyl group that are less recalcitrant for biodegradation. Other transformations are then catalyzed by lipase and esterase after the formation of carboxyl group or by endopeptidase for amide groups. Assimilation and mineralization. The formation of monomer does not guarantee their assimilation by microorganism. They have to use specific carrier to cross the cell wall or cytoplasmic membrane. Some monomer may stay in the surrounding of microbial cell without being assimilated. Inside the cell, the plastic monomers are oxidized through catabolic pathway to produce energy, cell structure and new biomass. Depending on the microbial abilities to grow in aerobic or anaerobic condition, there exist three essential catabolic pathways to produce the energy to maintain cellular activity, structure and reproduction. They are aerobic, anaerobic, respiration and fermentation. The assimilation refers to the integration of atom inside the microbial cell, but the degradation of the monomer may not be completed. The assimilation results in numerous secondary metabolites that can be transported outside the microorganism. They do not have the metabolic capability to transform in, or that do not need or metabolize or store it. The secondary metabolite excreted may be used by another cell that can perform further degradation. The mineralization refers to the complete degradation of primary and secondary metabolite that result in the excretion of completely oxidized metabolites such as carbon dioxide, nitrogen, methane, and water. Several factors for a better biodegradation of polymer plastic. By using surface active agent or including microorganism to produce surfactant to allow better attachment of microorganism on the polymer surface. Blending of the polymers with biodegradable synthetic polymers such as polylactic acid or polycarbolactam. Pre-treatment of the plastic, which include thermal, UV, high energy radiation and chemical treatment. Culturing those microorganisms that can efficiently degrade the plastic, that we called as a bioaddition of organism. Also, addition of nutrient may be limiting in the environment, that we called as a biostimulation. Thank you. Like, share, comment and subscribe for more videos.